Hey everyone, in today's video I want to talk about the electron transport chain, or ETC as it's often abbreviated. So I started out with this sort of cartoonish figure. Well, let me orient you to the figure here. So what we're looking at are the two membranes of the mitochondria. So these are the outer and inner membrane. So that's represented kind of by these pink little dashes. So this is a lipid bilayer. And then there's a space between the inner and outer mitochondrial membranes. And then this section above the figure here, this would be the cytoplasm. So in the cytoplasm, recall that glucose will be converted to pyruvate through the process of glycolysis. So the innermost chamber now, this is called the matrix. Okay, it's where Keanu Reeves and all his buddies hang out when they film that famous movie, The Matrix. Just kidding, that's an insider joke. But okay, so here we have the matrix. Now, recall now that pyruvate has to go through the pyruvate dehydrogenase complex. Okay, it's going to again get deposited as acetyl-CoA. That's then going to enter into the Krebs cycle. So we get two turns of the Krebs, and ultimately we're going to get 6 NADHs. We're going to get 2 FADH2s out of that, 2 turns of this, and then we get 2 GTP/ATP molecules. All right. So we covered that in a previous video, what was happening in the Krebs cycle. But ultimately then, what do those compounds do? What do these NADHs and FADH2s do? Well, that's where this transport chain comes in. So imagine we've taken now a cross-section of a mitochondrial membrane, and we're focusing in on the 4 main respiratory complexes. So they're just abbreviated as complex 1, complex 2, these are just Roman numerals, complex 3, and complex 4. And these are what are called integral membrane proteins. They're embedded in the inner mitochondrial membrane. But envision again, this is a surface of a membrane, so there are literally hundreds of thousands of these embedded all across the inner mitochondrial membrane. We're just focusing on these 4. There are also a couple of what are called mobile electron carriers. This one labeled Q, stands for coenzyme Q. It basically is going to flit around on the surface of the mitochondrial membrane and pick up electrons from complex 1 and 2 and dump them off to 3. We'll show that in a second. And then there's also another small mobile carrier called cytochrome C, and it's going to be involved in transporting electrons from complex 3 to complex 4. All right, so getting started now with how this process actually works. NADH is going to dump its electrons off to complex 1, creating NAD+. All right, so when we say dump its electrons off, it literally is taking this hydride with its 2 electrons, and it's reacting with a metal center that's present in complex 1. And so those 2 electrons are now going to go and reside on the metal center. And as that happens, the electrons are literally going to be flowing through this complex towards coenzyme Q. So I'm going to show the movement of these 2 electrons with sort of a dark, we'll sort of use a pink arrow here. So ultimately, those electrons are now going to go to Q. And along the way, at this particular step, a proton gets pumped. So we're going to take H+ here, and we're going to pump it right there. Okay, so what's going to happen is as these electrons flow through complex 1, a proton is literally moved from the matrix into this inner mitochondrial membrane space. So let's just circle that H+ right there. Okay, so coenzyme Q now is going to bring those 2 electrons now. It's going to bypass complex 2. We'll get back to complex 2 in a second. And it's going to dump those electrons off now to a metal center in complex 3. All right. Next, as complex 3 encounters a cytochrome C, it's going to then dump electrons off to cytochrome C. Cytochrome C is now going to dump its electrons to complex 4. Now, at each of those stages, complex 3 is going to take another proton and pump it into the inner membrane space. But that's only going to happen when complex 3 now gives its electrons to cytochrome C. So it actually really happens at this interface. Also, when cytochrome C dumps its electrons off to complex 4, another proton is pumped. Okay? So ultimately, each NADH that is in the matrix here, when it encounters complex 1 and dumps its initial electrons off, we'll just write this down here. Each NADH pumps 3H+ into the inner membrane space. Now the very final step, and I'm going to write this in pink here, these electrons need to ultimately go somewhere. All right, and what they're going to do, they're going to be involved in the conversion of oxygen into water. Okay, so oxygen is the ultimate electron acceptor of the electron transport chain. Notice they started over here. These two electrons are going to come over, go through this whole process, and ultimately it's at complex 4 where oxygen is now reduced into water. So oxygen is the ultimate electron acceptor, and this helps explain now why we need oxygen to do oxidative respiration. This is the place where oxygen is used. All right? So again, each NADH dumps its electrons off, we pump 3 protons, and we reduce an oxygen into water. So that's what happens for each of the NADHs. Now what happens with the FADH2s that are present or are synthesized as part of the Krebs cycle? Well, those interact with complex 2. So one easy way to remember that is FADH2, it has a 2 in the name, is going to interact with complex 2. So we could envision those coming over and actually reacting right there. So I'm just going to go ahead and draw that. Okay, and then those are going to reconvert back to FAD. So when those electrons are dumped off from the 2 hydrogens here, there again are going to be 2 electrons. Okay, and those are going to now flow and be picked up by coenzyme Q. So coenzyme Q is a unique protein. It can accept electrons from complex 1 or from complex 2. Okay. Now once the electrons have entered into the pathway here from complex 2, notice that the pathway now is exactly the same as it was before. So the result of this is that each FADH2 that enters the electron transport chain, it misses out on proton pumping at complex 1. So if we summarize here, each FADH2, it's only going to pump 2 protons, the one at complex 3 and complex 4. It misses out on complex 1. Okay, but ultimately those electrons then flow through from FADH2 just like they did with NADH, and they're going to convert oxygen into water. Okay, so this is what's called the electron transport chain. A major byproduct of these electrons flowing through is the creation of these pumped protons, and we call this now a proton gradient. So there are going to be more protons in the inner membrane space than there would be in the matrix. If you were to take a little micro pH probe, let's say, and you could somehow stick it into the inner mitochondrial space, and you measured the pH, because the protons are building up more here, this would be a little bit more acidic than the matrix would be. So ultimately, it's these protons that get pumped, this proton gradient now, that's going to now interact with the second half of this pathway that's going to ultimately make a lot of the ATP through what's called oxidative phosphorylation, and that's going to involve a new and different enzyme complex we'll cover in a separate video called ATP synthase. All right, so when it comes to picturing this, I would try and have a good picture of this in your mind, where it's all happening in the cell. Again, glycolysis and all that happens in the cytoplasm. But once those carbons from pyruvate get into acetyl-CoA, we go through the Krebs cycle. All of this is now happening in the matrix or on the inner mitochondrial membrane. Okay, so that covers the ETC. I hope that helps.